Good morning, friend. Today we are going to discuss immunofluorescence and enzyme linked immunosorbent assay. So, first, we are going to discuss the immunofluorescence. So, before starting the lecture about immunofluorescence, I want you to introduce the term that is fluorescence. So, what exactly the meaning of fluorescence? So, fluorescence is the property of certain dyes which absorb the rays of one particular wavelength and emits the rays with a different wavelength. What does it mean? Actually, there are some dyes which absorbs the wavelength in a UV range. That is basically they absorbs the ultraviolet rays and they emit the wavelength in a visible range. So ये चीज आपको पता होगी कि जो भी हमें कलर दिखाई देता है, सपोज करो the leaf of a plant are suppose that they are appearing green in color. What does it mean? कि they are emitting the green wavelength. That's why they are appearing in green in color. Get it? तो वैसे ही डाइस के साथ में होता है, वो किसी दूसरे वेवलेंथ को एब्जॉर्ब करते हैं, दैट इज यूवी रेज को, एंड दे आर एमिटिंग द वेवलेंथ इन अ विजिबल रेंज, सो दैट इज कॉल्ड द फ्लोरोसेंस. अब अगर जो अलग सपोज दैट विजिबल रेंज में आपके पास कहाँ तक रेंज होती है, फ्रॉम 400 टू 700 नैनोमीटर. सो जिस भी वेवलेंथ की लाइट को वो ओमिट करेंगे वो कलर आपको दिखाई देगा. यू कैन सी इन द डायग्राम ऑल्सो कि यहाँ पे आपको ग्रीनिश ब्लू कलर दिखाई दे रहा है, रेडिश कलर दिखाई दे रहा है. क्यों? क्योंकि इन दोनों ने क्या किया है, यूवी रेज को एब्जॉर्ब किया और डिफरेंट वेवलेंथ को, बट इट इज इन दी विजिबल रेंज ओनली, एमिट किया है. फॉर एग्जाम्पल फ्लूरोसिन आइसोथायोसाइनेट, ये क्या करता है, दिस इज अ बेसिकली फ्लोरोसेंट डाई विच एब्सॉर्ब्स द यूवी रेज and emit the blue-green wavelength. What about the lysamine, rhodamine? This also absorbs the UV rays and emit the orange-red wavelength which is in the visible range. So इसके अलावा भी बहुत सारे डाइज है और ये जनरली दो डाइज है जो इम्यूनोफ्लोरेसेंस में यूज किए जाते हैं. So now there are two types of tests, that is one direct fluorescence test and another one is indirect immunofluorescence test. Now we are going to discuss that. So first is your direct immunofluorescence test, in which what you have to do, first you have to take the slide and then you have to add the antigen. Okay, what you have to do, you have to antigen. एंटीजन मीन्स व्हाट, अगेंस्ट विच वो एंटीजन आपको लेना है. सपोज दैट आई एम सफरिंग फ्रॉम अ डिजीज, तो मेरे सैंपल के अंदर वो डिजीज कॉजिंग जो एजेंट होगा, पैथोजन होगा, वो प्रेजेंट होगा. सो व्हाट यू हैव टू डू, फर्स्ट यू हैव टू टेक दिस स्लाइड एंड यू हैव टू ऐड माय सीरम. तो सीरम में क्या प्रेजेंट होगा, दैट स्पेसिफिक एंटीजन. अगर इफ देयर विल बी अ इन्फेक्शन देन ओनली स्पेसिफिक एंटीजन इज प्रेजेंट, एंड वी आर अज्यूमिंग दैट कि मुझे इन्फेक्शन है, सो इन्फेक्शन है तो मेरे सीरम में क्या होगा, एंटीजन होगा. तो यू हैव टू ऐड माय सीरम. After that, what you have to do, you have to add the fluorescent labeled specific antibody. Specific antibody means what? Specific antibody किसके अगेंस्ट होगी, जिस एंटीजन को आप डिटेक्ट करना चाहते हैं, what type of antigen you want to detect, उसके अगेंस्ट एंटीबॉडी होगी, एंड यू हैव टू लेबल दैट एंटीबॉडी विथ अ फ्लोरोसेंट डाई. तो आपने क्या रेडी कर लिया, फ्लोरोसेंट लेबल्ड स्पेसिफिक एंटीबॉडी ऐड किया. आफ्टर दैट आपको क्या करना है, देन यू हैव टू ऑब्जर्व द स्लाइड अंडर यूवी रेज. Why under UV rays? Why? Because we have studied that जो fluorescent dye होती है absorbs the UV rays and emit the wavelength in a visible range. So जब आप UV rays में रखेंगे तो fluorescent dye जो होगी it will absorb it and emit. अगर आपको कोई कलर दिखाई दे रहा है, कोई भी जो भी आपने यूज किया, सपोज करिए आपने रोडामीन ऐड की है डाई, सो अगर आपको ऑरेंजिश रेड कलर दिखाई दे रहा है, दैट मीन्स द टेस्ट इज पॉजिटिव. वाई टेस्ट इज पॉजिटिव? बिकॉज जो फ्लोरोसेंट लेबल्ड एंटीबॉडी था वो स्पेसिफिक एंटीजन में, एंटीजन जो कि सीरम में प्रेजेंट था, उससे बाइंड किया है और तभी यू आर एबल टू सी द फ्लोरोसेंस. दैट्स व्हाई the test is positive if you able to see the fluorescence. Get it? This is the direct immunofluorescence. But there is a one drawback of direct immunofluorescence test, that is, for every antigen, as we know that antigen are specific for its antibody, so for every type of antigen you require different fluorescent labeled antibody. अभी हर एक चीज को टेस्ट करने के लिए हर एक एंटीबॉडी को लेबल करना क्या है, पॉसिबल नहीं है, इट इज वेरी कॉस्टली. सो टू अवॉइड दैट एक और टेस्ट आया, दैट इज द इनडायरेक्ट इम्यूनोफ्लोरेसेंस टेस्ट. क्लियर है? यहाँ पे व्हाट यू हैव टू डू, हमने यहाँ पे क्या किया, स्लाइड में नॉन एंटीजन लिया. व्हाट यू मीन बाय नॉन एंटीजन, यानी जो हम चेक करना, सपोज करिए मुझे सिफिलिस का इन्फेक्शन है, तो मुझे एंटीजन कौन सा लेना पड़ेगा, you are checking that my serum contains antibodies against syphilis or not. You have to check कि मेरे बॉडी के अंदर सिफिलिस के अगेंस्ट एंटीबॉडीज प्रेजेंट है या नहीं है. तो है या नहीं कैसे चेक करोगे, उसके लिए यू रिक्वायर्ड अ सिफिलिस का एंटीजन, सो दैट्स व्हाई इट इज कॉल्ड द नॉन एंटीजन. यहाँ पे स्लाइड में यू हैव टेकन द ट्रेपेनोमा पैलिडियम, दैट इज द एंटीजन ऑफ सिफिलिस. Okay, now you have to add the patient serum which contains the antibody. You have to take my serum which contains antibody. Now after adding this, what will happen, antigen will bind with the antibody. Now after that you have to add the fluorescent labeled antibody, and this fluorescent labeled antibody will bind to the primary antibody with the help of FC region. Look, I told you that antibodies में क्या होता है, एक FAB region होता है through which antibodies bind with the antigen, और एक FC region होता है that is constant in every type of antibody. तो यहाँ पे जो आपने fluorescent labeled antibody यूज किया है, that antibody can bind with the primary antibody with the help of FC region. Why? Because FC region is constant in any type of antibodies. गेट इट? तो यहाँ पे आपको फ्लोरोसेंट लेबल सिर्फ एक ही टाइप की एंटीबॉडी को करना पड़ेगा, क्योंकि एफसी रीजन तो कोई भी एंटीजन हो, किसी के भी अगेंस्ट एंटीबॉडी हो, एफसी रीजन विल बी सेम. सो आपको यहाँ पे एक ही टाइप की फ्लोरोसेंट लेबल एंटीबॉडी रेडी करनी होगी, दैट विल बाइंड विद द प्राइमरी एंटीबॉडी. प्राइमरी एंटीबॉडी कहाँ से आई, दैट एंटीबॉडी केम फ्रॉम द सीरम, पेशेंट के सीरम से आई. गेट इट? सो कहाँ पे थे हम कि हमने व्हाट वी डिड, हमने ऐड किया फ्लोरोसेंट लेबल एंटीबॉडीज. Now again you have to visualize the slide in the presence of UV rays. If you'll observe the color, अगर आपको कलर दिखाई दे रहा है ऑरेंज या जो भी आपने डाई यूज किया है, अगर कलर दिखाई दे रहा है, दैट मीन्स वॉज कि डाई फ्लोरोसेंस शो कर रही है. डाई फ्लोरोसेंस शो कर रही है, व्हाट डज इट मीन कि डाई वहाँ पे डाई वाली जो एंटीबॉडी है दैट इज बाइंड विद द प्राइमरी एंटीबॉडी. और प्राइमरी एंटीबॉडी कहाँ से आई, पेशेंट के सीरम में से आई है. तो दैट मीन्स व्हाट, इफ यू ऑब्जर्व द फ्लोरोसेंस, दैट मीन्स test is positive, patient is suffering from the particular disease. Get it? And here just remind this, after each and every step you have to do the washing. Why washing? Because washing removes the unbound antigen. Get it? Washing removes the unbound antigen and antibody. Suppose करो यहाँ पे आपने फ्लोरोसेंट एंटीबॉडी ऐड किया, अगर आपने वॉशिंग नहीं किया, इन दैट केस क्या होगा, फ्लोरोसेंट बॉडी तो उसमें प्रेजेंट ही रहेगी ना, आपके सिस्टम में सैंपल में रहेगी, वो थोड़ी भले वो बाइंडेड फॉर्म में नहीं है, बट इट इज प्रेजेंट देयर ओनली ना. सो इफ इट इज नॉट बाउंड यू हैव टू रिमूव इट. Get it? How will you remove it? By washing. If you do the washing, तो अगर suppose that for example patient के serum में antibodies नहीं है, in that case what will happen, if you'll add the known antigen and if you'll added the patient serum which doesn't contain any type of antibody, then when you will add the fluorescent label antibody, it will not bind with the primary antibody. Why? Because there is no primary antibody present in the patient serum. Get it? So after washing what will happen, fluorescent antibody will removed, and when you will see the slide in the UV rays, you are not able to see any kind of fluorescence, क्योंकि fluorescent labeled antibodies are already removed. So after each and every step, you have to do the washing just to remove the unbound antigen or antibodies. Get it? I hope it is clear to you ki what is direct immunofluorescence test and what is indirect immunofluorescence test, and why we are using indirect immunofluorescence test. What is the drawback of direct immunofluorescence test? Now we can see the application of both tests. For example, direct immunofluorescence can be used for the identification of bacteria, viruses, or other antigen, and it is routinely as a sensitive method for diagnosis rabies by detecting the rabies virus antigen in the brain smear. You can detect the rabies virus in the brain smear also. And what basically rabies virus means, you can detect the antigen in the brain smear. Of which antigen? Rabies antigen, with the help of direct immunofluorescence test. And in case of indirect immunofluorescence, it can be used for the diagnosis of syphilis. Okay, syphilis के diagnosis के लिए you can use this, in which you have to use fluorescent treponemal antibody test that I have already explained you. Second one, indirect immunofluorescence test. Get it? Now we are going to proceed. That is our next technique, enzyme linked immunosorbent assay, that is called ELISA. So ELISA is a quantitative immunological procedure in which the antigen antibody reaction is monitored by the enzyme measurement. Actually, it is a plate-based assay technique designed for detecting and quantifying substances such as peptide, proteins, antibodies, and hormone. So what does it mean? With the help of ELISA, even you can detect the antibodies, you can detect hormones, peptides, and protein also. And it is basically a quantitative immunological procedure. Quantitative means you can detect the presence or absence, as well as you can detect the amount of substance involved. That's why it is called the quantitative immunological procedure. And in this test, the antigen antibody reaction, basically it is monitored by the enzyme measurement. How that I am going to explain you with the help of diagram. Get it? And it involves the detection of analyte. That analyte is what? Basically that you are measuring quantitatively or qualitatively. Okay, so basically it is the procedure used for the detection of analyte. Analyte may be anything, peptide, protein, antibodies, hormone, anything that you want to analyze qualitatively or quantitatively. Now, what are the enzyme used in case of ELISA? So there are a lot of enzyme used, but I am just giving you some example. For example, horseradish peroxidase, alkaline phosphatase, catalase. These are the enzymes which are generally used in case of ELISA. Now, these are the basic steps that involve in ELISA that I'm going to explain you with the help of diagram. Look at this. This is an example of sandwich ELISA. I hope हमने जीवन में sandwich खाया होगा. 2 ब्रेड बीच में आलू का मसाला. Here also. यहाँ पे भी 2 ब्रेड act as an antibody and आलू का मसाला act as an antigen. Get it? That's why it is called the sandwich ELISA. Why it is called the sandwich ELISA? Because aloo ka masala is between the 2 bread. That means antigen is sandwiched between the 2 antibodies. Get it? That's why it is called the sandwich ELISA. Now we are going to discuss the procedure of sandwich ELISA. So first you have to take the micro titer plate in which you have to immobilize the antibody. What you have to do, आपको एक micro titer plate लेना है and then you have to do the immobilization of antibody. After that you have to add the patient serum. Patient serum जिसमें आप एंटीजन का प्रेजेंस चेक कर रहे हैं. ओके, सो पेशेंट का सीरम आपने लिया है तो पेशेंट के सीरम में व्हाट यू आर चेकिंग दैट द पर्टिकुलर पेशेंट इज सफरिंग फ्रॉम अ पर्टिकुलर डिजीज और नॉट. तो आपने पेशेंट का सीरम ऐड किया. सपोज दैट पेशेंट इज सफरिंग फ्रॉम अ पर्टिकुलर डिजीज. सो 100% चांसेस कि उसके ब्लड में, उसके सीरम में आपको एंटीजन मिलेगा. सो एंटीजन विल बाइंड विद द immobilized antibody. After that, you have to add the enzyme linked antibody. Now I told you this जब हमने when we studied the general features of antigen antibody interaction. I told you कि antigen has lots of valency. Get it? It doesn't mean कि सिर्फ antigen के पास में एक single epitope नहीं होता है. There will be a chance of multiple epitope present on the antigen. So एक epitope जो main epitope है उसके थ्रू वो क्या है, immobilized antibody से bind है. गेट इट? तो आप दूसरा एपिटोप यूज कर सकते हैं. यू नो द एंटीजन ना, यू नो सपोज दैट एलाइजा जनरली यूज फॉर डिटेक्टिंग द HIV वायरस. ओके, सो HIV वायरस के सपोज दैट 2 एपिटोप है. तो एक एपिटोप से तो वो इमोबिलाइज्ड एंटीबॉडी से बाइंड है और दूसरे एपिटोप के अगेंस्ट आपने क्या किया हुआ है, एंजाइम लिंक्ड एंटीबॉडी डिजाइन किया है. So with the help of one epitope, it is bind with the immobilized antibody, and with the help of another epitope, it will bind with the enzyme linked antibody. Get it? So after adding the antigen, you have to add the enzyme linked antibody that will bind with the second epitope of the antigen. Now what will happen? Then you have to add the substrate. Why? Because it is enzyme. So enzyme convert the substrate into the product. So substrate will convert it into the product and it will show some color. So if you're able to see the color, that means test is positive. Person is suffering from the particular infection. And here also, after each and every step, you have to do the washing just to remove the unbound antigen or antibody. For example, in first step, after immobilization of antibody, you have to do the washing just to remove the unbound antibodies. And after adding the human serum which contains antigen, after that also you have to do the washing just to remove the unbound antigen. And then after adding the enzyme linked antibody, again you have to do the washing just to remove unbound enzyme linked antibody. Okay, and after adding this substrate, there is no need to add the there is no need to do the washing. Okay, after adding the substrate, if the enzyme linked antibody will be present, then only it will convert the substrate into product and you are able to visualize that product. ठीक है, अगर आप विजुअलाइज कर रहे हैं मींस एंजाइम लिंक्ड एंटीबॉडी प्रेजेंट है. वो क्यों प्रेजेंट है? क्योंकि एंटीजन प्रेजेंट है. एंटीजन कहाँ से आया? आपके सीरम से आया. So test will be positive. Yani person is suffering from some infection. Get it? Even you can see here, suppose that कैसे काम किया जाता है? Horse radish peroxidase. What you have to do? You have to add the basically hydrogen peroxide. This will convert it into H2O and nascent oxygen with the help of horseradish peroxidase, and this nascent oxygen will then convert the diaminobenzidine into the oxidized diaminobenzidine. So this diaminobenzidine is colorless. So this substrate is colorless. In the presence of enzyme which converted the hydrogen peroxide into the nascent oxygen. So nascent oxygen then convert the substrate into the product, that is the colored product which shows the brown color. So if you are able to visualize the brown color, that means test is positive. Get it? Now there are different types of ELISA. Look at here. Capture assay, that is the sandwich ELISA that I have already explained you. Immobilized antibody, patient serum, antibody यहाँ पे, and you can use another enzyme linked antibody also. Then you have to add the substrate that will convert into the product. What about the indirect assay? In indirect assay, what you have to do, you have to just add the known antigen. ठीक है, known antigen in the microtiter plate. Fine. After that, you have to add the patient serum, which contains the primary antibody. Then again, you have to add the secondary antibody, enzyme linked secondary antibody, which will bind with the primary antibody with the help of Fc region. Now what will happen when you will add the substrate, then the substrate will convert it into the product and you will able to observe some color. Get it? In case of direct assay mein what you have to do, you have to add the antigen and then you have to add the primary antibody which is conjugated with the enzyme. So in this case you can directly observe the color if antigen is present in the serum, patient serum. Get it? And just please remember this, that after each and every step you have to do the washing just to remove the unbound antigen and or unbound antibodies. So these are three different type of ELISA. Sandwich ELISA, in which antigen is sandwiched between the two types of antibody, and indirect ELISA, you have to take the antigen, then primary antibody, then secondary antibody linked with the enzyme. And in direct ELISA, just you have to take the patient serum which contains the antigen and you have to add the enzyme linked primary antibody. After that substrate add karna hai, then substrate will convert into the product with the help of enzyme and you can observe the color. Get it? So this are the types of ELISA. Now what are the application of ELISA? Actually it has a huge application. Basically the screening donated blood for evidence of viral contamination by HIV-1 and HIV-2. Okay, so you can detect the presence of anti-HIV antibodies. What does it mean? कि आप anti-HIV antibodies detect कर सकते हैं with the help of ELISA, and you can check the presence of antibodies in the serum. किसके antibodies? Hepatitis C के against antibodies you can check. Even testing of both antibodies and viral antigen of hepatitis B. Even you can check the hepatitis B antigen as well as the antibodies against the hepatitis B in the patient serum. You can check both antigen and antibody in case of hepatitis B. What type of antibodies? Antibodies against the hepatitis B virus. Get it? And even it is used for measuring the hormone level. For example, HCG hormone as a test of pregnancy. Pregnancy में HCG hormone level measure किया जाता है. That also you can detect with the help of ELISA. As I told you in the starting that ELISA can be used for detection of protein, peptides, hormone, antibodies. So the presence of hormone, HCG hormone, can also check by the ELISA. And even you can check the LH hormone, that is the luteinizing hormone, determining the time of ovulation. You know that today now in market the ovulation kit is also available, जो आपको ये बताता है कि when is your fertile days, so कब आपका ovulation होने वाला है, that is detected by the kit. So that kit is based on the ELISA only. Get it? Now even you can check the thyroid stimulating hormone, that is TSH, T3 hormone and T4 hormone for the thyroid function with the help of ELISA. Get it? So these all are the application of ELISA. So what we have studied today, we have studied the immunofluorescence and their application. Then we have studied the ELISA, that is enzyme linked immunosorbent assay, and their applications. Now I think you can understand why ELISA is called enzyme linked immunosorbent assay. Why? Because the antigen or antibody, whatever you are determining, that is monitored with the help of enzyme. That's why it is called the enzyme linked immunosorbent assay. Get it? So this is all about the immunofluorescence and ELISA. Thank you.